Good afternoon, everyone. Good afternoon, Anuj, Rishita. So, let's begin with today's introduction session. Hello, everyone. Is my screen visible and am I audible? Yes, okay. Can we begin with today's session? Please let me know. Can we begin? Yes? Okay, let's start. Welcome everyone to the NPTEL course on novel technologies for food processing and shelf life extension. And Professor H.N. Mishra is a course instructor. He is from the Agriculture and Food Engineering Department, IIT Kharagpur. I am Shikha Sharma. I am a research scholar in the Department of Chemical Engineering, IIT Guwahati. This is our last uh, interaction session for week 12. Uh, yeah. So the topics which we will be discussing in today's session will be functional foods and nutraceuticals, algae-based health foods, gluten-free breads and pasta, food powders and premixes, GMP and GHP in food industry. And in the last session, each uh, topic will be having one lecture video which is provided in week 12 content. And also there is sixth video which is regarding course summary and FCTL facility in the IIT Kharagpur. So in total six lecture videos are there for this week 12. So I hope you have already gone through six lecture videos. And if you have any doubt regarding today's session, because our session will be based on these topics, so you can ask your doubts based on these six topics. And if you have any doubt in my questions or the problems which we'll be solving today, in the concepts or in the problems, you can please ask. So, without wasting much time, let's just begin with today's first question. Which among the following statements is are true regarding functional foods? So, we have functional foods which provides extra nutrients to the people who are not having those kind of nutrients. So, out of these four statements, which statement is true for functional foods? So, first statement is designed to provide health benefits to consumers. Statement two is foods whose inherent health benefits go beyond basic nutrition, including the prevention of disease and the promotion of wellness through nutrition. Statement 3 is similar in appearance to conventional foods and are consumed as a part of the usual diet. Fourth statement is often are associated with bioactive compounds in small quantities like antioxidants, omega-3 fatty acids, phytochemicals, and probiotics. So we need to tell only one statement is true, only one and two, Only 1, 2, and 3, or all of them? 1, 2, 3, 4. Which is correct?
All of them, ma'am.
All of them?
Yes, ma'am.
Okay. So
Option
we got
E.
two answer option.
D.
Option D. We got two answer as option D. Anyone is having any other answer? D. All are having D answer. Many people are saying D. All even. Okay. Let's discuss the solution. D is correct. All of them are true in regards to the functional foods. So functional foods, they are basically, obviously they are designed to provide health benefits to the consumers. Even they are the foods whose inherent health benefits go beyond basic nutrition, including prevention of the disease and the promotion of wellness through nutrition. They are similar in appearance to conventional foods and are part of the usual diet. That's why we will have those kind of functional foods. If they are different from the conventional foods and their taste is not, or the sensory property is not similar to the functional foods, then why we will going to eat? So it is it is obvious that whenever we are designing or developing any functional food, we should make it as similar as conventional food, and which is having higher health benefits than conventional food. That's why then only customers will going to take that or buy that food. These are often are associated with bioactive compounds in small quantities. So they can have antioxidants, omega-3 fatty acids, phytochemicals, and probiotics. That's why they are having higher functionality than the conventional food. So option D is correct, as you all said. So is this clear to everyone, right? So we are moving to the next question. Second question, which among the following are the correct sequential steps in food product development and engineering? So we have four steps: concept development, availability of engineering resources, identification of opportunities, and then testing, market, production, and national production. So we need to tell which is the correct step, which step we will going to take while we are making product, or and what which step which we will going to follow at the at the later stages. So usually in the product development, basically this concept of product development or new product development, it mainly encompasses the entire process of thinking, creating, planning, and even commercialization. So this the new product development is all about converting new and untried ideas into the sorry into workable products. So new means which we have not tried. So new and untried ideas, they will be converted into some workable products. That is the definition how in or what we do in the product development. And it is not just idea, it is the entire process from ideation to commercialization. So let's come back to the question, which is the correct step or the correct sequence of product development and engineering. Anyone?
Option C, ma'am.
Ma'am, B.
C, C,
Option B.
D, B or D?
B.
B, okay.
Then C.
C,
Yes, ma'am.
we are having a mixture of answer this time, B, C and D. No, okay, D is maybe previous one. So C, B, mixture of these answers we are having. Let's see which is correct. So C is correct. So the steps which are involved in the product development and engineering, it comprises of first we see availability of engineering resources, and then identification of the needs and opportunities, and then concept development, and then there will be testing of the developed concepts, and then testing of the product in the market production, and finally the large scale or national production. Usually in the product development, first we generate idea, which is based on like maybe from the brainstorming, we develop ideas, and then those ideas will be screened out based on the resources available, and then concept development and testing. So it is done usually by the experienced person, where the whole concept of the product is being developed, and after that marketing, when the product will be developed, we when we develop the concept, we have to think about what market strategy or business strategy we are going to follow, which includes mainly 4 P's. Like 4 P's means, here it includes product, about our product we think, then we can think of price, uh, place or positioning, and then promotion or the marketing. All these, while we developing the product, we must think all these things also, where we keep our product, like that, in all these 4 points. Then we develop the product, then small scale study or small scale production will be there, and then finally at the large scale market we send product. So option C is correct, not B. So please see that first is, we see the like after just ideation, we see the first we see engineering resources, like what are the resources we are having. On the based on that, we find the opportunities from those resources what we can make, and then based on that we develop some idea which is workable, which must be workable. Then we think we must test the product in the market production, small scale production, and then at the large scale, which is national production. So is this question clear to everyone?
Yes, ma'am.
Yes, let's move to the next question then. So it is a match the following question. So match the type of algae. So in column 1, we are having different algae, and in column 2, its characteristics are there and we need to match. So we are having 4 algae: cyanobacteria, rhodophyta, chlorophyta, and phaeophyta. So we need to tell which is like out of these which are having these characteristics, like eukaryotic algae and found in the freshwater lakes. And then second is the branched and filamentous thallus. And third is photosynthetic prokaryotics. And then fourth is unicellular and colonial flagellatas. So which
Option
option
C.
can be correct? Option C.
Yes, ma'am.
Okay. Any other option do we have? Option C, option D, either option C or D. Maximum but are saying option C. Let's see. Yes, C is correct. So blue-green algae, which is also known as cyanobacteria, are photosynthetic prokaryotics organisms. In red algae, which is also known as, this is the blue-green algae, first image. And then second, which is red algae. This is red algae. So it is also known as rhodophyta, and they are eukaryotics algae and are found in the freshwater lakes. Unicellular and colonial flagellatas, they are in the green algae or chlorophyta, which is green algae this. And then there is this brown algae, which have branched brown algae, which is known as phaeophyta, and it is having branch and filamentous thallus. So option C is correct based on this, and this explanation is provided in the lecture video also. So please go through this lecture where this uh, different algae are there and their characteristics are mentioned. So is this question clear? Can we move to next question?
Yes, ma'am.
Okay, thank you. Let's move to this question. Blank is an inflammatory autoimmune condition of small intestine and interferes with absorption of nutrients from food, which is triggered by gluten. Celiac disease, IBD, which is inflammatory bowel disease. C is diabetes mellitus, and fourth is Hashimoto thyroiditis.
A disease. A, option A.
Option A, ma'am.
Option A.
Yes, ma'am.
Okay, everyone is saying option A.
Yes, ma'am.
Let's see in the chat also. Yeah, most of the everyone is on the option A only. So yes, it's celiac disease. Everyone is right. So celiac disease is basically an inflammatory autoimmune condition of small intestine that damages the small intestine and interferes with the absorption of nutrients from food, triggered by gluten in genetically susceptible individuals. People suffering from celiac disease, they cannot tolerate gluten. So it is the normal intestine and it is celiac intestine when there is this intolerance, because in the small intestine it will not able to, the people who are having celiac disease, they will not able to digest gluten. They don't have that a kind of particular enzymes to digest or break down gluten protein chain, so they have that disease. Second is the inflammatory bowel disease, which is IBD. It is a group of inflammatory condition of the colon and small intestine, and it is having two disease in particular, which are Crohn disease and UC, which is ulcerative colitis. So these two disease, ulcerative colitis, which is mainly like the principal disease in this condition. So this condition, it involves inflammation and sores, which are called ulcers, along the lining of the colon and rectum. In Crohn disease, what happens? In this type of IBD, the lining of the digestive tract is inflamed. The condition often involves the deeper layers of the digestive tract. In this disease, what happens? Most commonly, small intestine is affected, and however, it also can affect large intestine and uncommonly the upper gastrointestinal tract. Symptoms of both UC, ulcerative colitis, and Crohn disease usually include belly pain, diarrhea, rectal bleeding, extreme tiredness, and weight loss. So this is IBD. And then diabetes mellitus. In this, it is basically a metabolic disease which involves inappropriate elevated blood glucose level. In the diabetes mellitus, it has several categories, like it includes type 1, type 2, then maturity onset diabetes of the young, and then gestational diabetes, then neonatal diabetes, which are there in infants of lesser than 6 months. So it is because of the genetic problems. And then secondary causes due to endocrinopathics, and then steroids use. So these kind of like diabetes is also having like many types. It can even from a small infant to a young or old people, it can happen. So mainly there are two diabetes, which are type 1 diabetes and type 2 diabetes. So which classically result from defective insulin secretion and or action. So in T1 type 1 diabetes, it presents in children or adolescents, and in type 2 diabetes is basically it affects middle aged people or older or adult people. So in type 2 diabetes, this is basically in middle aged or older people which are having prolonged hyperglycemia due to poor lifestyle and dietary choices. Last is Hashimoto thyroiditis. So this disease is basically an autoimmune disorder that can cause hypothyroidism or underactive thyroids. Rarely, this disease can cause hyperthyroidism or overactive thyroid. So in the image we can see this is a thyroid gland. So in thyroid gland, if it affects this disease. So the thyroid is the small butterfly shaped gland in front of your neck. In people with this disease, suffering from this disease, the immune system makes antibodies that attack the thyroid gland. So antibodies, they are attacking this thyroid gland. Large number of WBC, white blood cells, which are part of the immune system, it build up in the thyroid. The thyroid becomes damaged and cannot make enough thyroid hormone. So due to uh, uh, more of the antibodies going and attacking thyroid gland, it cannot produce thyroid hormones. And because thyroid hormones control our body uses energy, so they affect nearly every organ in our body, even the way our heart beats. So this is Hashimoto's thyroiditis. So from all these, only celiac disease match this column. So match this blank. So celiac disease is the answer. So is this clear to everyone? Can we move to next slide? Next question.
Yes, ma'am.
Okay, thank you. Let's move to the next question, which is about gluten-free bread. So development of gluten-free bread from batters results in better water retention ability during baking, functionality of gluten, gas retention capacity, improved crust and crumb structure.
Option D.
Option
Option
D.
D. Slide is not visible. Slides are not visible. Is this the slide visible?
Yes, ma'am. Now it's visible.
Yeah, I have shared on the screen. So when I am entire screen, so if I am using some other application, it is visible. The slides are not visible at that time only. Otherwise, slides are visible. D, C, D. Mostly D and C. C and C, right? Gas retention capacity and improved crust and crumb structure. So answer is C, gas retention capacity. As gluten-free bread preparation, in that we know that gluten-free flour we take, rice flour mainly, and in that water, salt, sugar and hydrocolloids are being added. The hydrocolloids and yeast, they act as an improver. Act as an improver. After a very short mixing of about maybe 2 minutes, around 500 gram of the formulation is taken and put in a pan at 30 degrees Celsius and 85% relative humidity for 30 minutes. These conditions help yeast to act, and finally it is baked. Lastly, the baked bread is cooled and packaged. So this is how we prepare gluten-free bread. So in this, basically rice flour is given some microwave treatment to modify its starch. So we are giving in this condition rice flour some kind of treatment like microwave treatment to modify starch. So the microwave treatment of rice flour provides improvement in its functionality of the material. So as per this, this diagram which is provided in the lecture video also, that this first case where there is a modification of rice flour by microwave treatment, it provides functionality of the gluten, which is functionality of gluten. This okay, so it is because of rice flour treatment and modification of the rice starch. And then second is the some citric fiber, it is mixed to improve the water retention ability of the batter. So during baking, so for that we are adding citrus fibers, which is option A. So for this we are adding citric fiber. And then A unique combination of hydrocolloids and modified starch, which is partially gelatinized rice flour, is added to the formulation for structure binding. This formulation gives an improved crust and crumb structure, and finally it is baked under usual conditions like those in the normal bread. So for improving crust and crumb, we are adding combination of hydrocolloids and modified starch. So for this option D, we are adding hydrocolloids and starch, modified starch. And for option C, which is our correct answer, so in this process of development of gluten-free bread, there is a preparation of bread from batter instead of dough. So because we are making batter instead of dough, so there is higher gas retention. So option C is correct because development of gluten-free bread from batter, so we have more gas retention. Is this question clear?
Yes, ma'am.
Yes, let's
Yes,
move to next question then. Yes, okay. Which of the following statements is wrong? The basic GMP program is not a prerequisite of HACCP accreditation. The TQM process involves quality control, quality assurances, and quality improvement. Then C statement is monitoring of CCP comes under preview of HACCP. Option D is statistical analysis is an integral part of quality analysis of food products. So we know that HACCP, which is a food safety tool, it stands for H means hazard, A means analysis, C means critical, another C is control point. So hazard analysis, critical control point. So hazard is any food contamination or spoilage. In this, A stands for finding out the source of contamination or the hazard. And then critical means the level of danger. After particular level or particular concentration, component can become toxic. So it can be dangerous for human. Control is the remedial actions. Point means the stage for corrective action. So this is HACCP. So we need to tell which is wrong statement for HACCP.
option A, ma'am.
Option
Option
A.
A. Everyone, option A. So let's see. Answer is option A only. Let's understand some basic concepts also. So HACCP is basically a preventive approach to food safety that identifies potential hazards and implements control measures at critical points in the production process. So HACCP, it has basically 5 steps. These are the 5 steps in HACCP, and there are 7 principles in the HACCP. So it is given by Codex Alimentarius that when so its 5 steps involve forming a HACCP team, and then product description, identify the intended use, creating a manufacturing process list, and on-site confirmation of manufacturing process list. These are the 5 steps or the 5 preparatory steps for hazard analysis. And then there are the principles, 7 principles or hazard factors analysis HACCP plan formulation. So these 7 principles are being applied. So in total 12 points are there in HACCP or HACCP system. So first is analysis of hazard, and then there is the determination or determining important control points or CCP, and then setting management standards, and then setting the monitoring methods, and setting the improved measures. After that, setting of the verification method, and then keeping the records or documents. And what is CCP? Critical control point. It is a point or a step or a procedure at which control can be applied in a food system or where the food manufacturing system is going on. So there will be some control points in the whole process to understand that the product to determine or to maintain the food safety. So some step or the points or the procedure, they can be applied to control or prevent or eliminate hazard. What is We are discussing all the terminologies which are mentioned in the question first. So HACCP we already discussed, and then CCP we discussed. Now let's discuss what is TQM. So TQM, which is total quality management, is basically a holistic approach to a long-term success through customer satisfaction. It basically involves all the members of an organization. It is organization participating in improving processes, products, service, and the culture in which they work. So it is holistic. In the holistic system, there should be it is saying that there should be continuous improvement. So in the context of food safety, TQM aims to ensure that every step of the production process meets high standard of quality and safety. So there should be continuous improvement, and there should be high quality and safety in food products. Next is GMP. What is GMP program? The GMP stands for Good Manufacturing Practices. In GMP, it involves the practices required to conform to guidelines recommended by agencies that control the authorization and licensing, which is FSSAI, set some guidelines for good manufacturing practices. So that needs to be fulfilled. So authorization and licensing of manufacture and sales of products. So it involves hygiene, sanitation, process control, quality control, and documentation. So this is GMP, and every industry, food industry must follow all these points. So coming back to our question, that which of the following statement is wrong? So first statement is wrong because basic GMP program is prerequisite of HACCP accreditation, not is it's not it is a prerequisite. uh, it is not a it's saying not a prerequisite, which is incorrect. It is a prerequisite. And the second statement, which is TQM, it is basically includes quality control and quality assurance and quality improvement, which is correct. Then third statement is monitoring of CCP comes under preview of HACCP, as we seen here in step 7, that determining important CCP. So this statement is also correct. And then statistical analysis is an integral part of quality analysis of food products. So, which is also correct. We have to do statistical analysis because there should be in points given to each step, and then for calculating we need to do statistical analysis. So, is this question clear? Can we move to next question?
Yes, ma'am.
Yes.
Yes,
So
ma'am.
next, yes, okay. Let's move to this question. The HACCP team is not responsible for which of the following jobs? Updating the HACCP plan annually or as processing changes occur. B is checking CCP documentation each hour. C is submission of reports to the top management. D is drafts the policies fulfilling the statutory and regulatory requirements.
Ma'am, B.
B,
C. C.
B and C. Let's see everyone's answer. So, D, 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 B, D, D and B. So, let's see. D is correct. It is not responsible for by the HACCP team. Basically, HACCP, which is Hazard Analysis Critical Control Point, it basically is an important aspect of GMP, and it can be a part of GMP, and it is a systematic program to ensure food safety in a industry. So the members of GMP team, it consists of people with multidisciplinary knowledge and experience, which drafts the policies, fulfill the statutory and regulatory requirements. So they drafts the policy, and this option D is correct because they are doing this, and then maintain the integrity of the policy, then conduct proper reviews, submit reports to the top management, which is also correct, submission of the report, and then and maintain proper communication. So, and update HACCP plan because they make plan and annually, and they like they note also the changes, but each hour they like checking CCP documentation each hour is not possible, so it is incorrect. So in the if we see in the diagram also, the organization's role is that there should be setting of the organization, it must set up a GMP team, and then there should be the formation of SOPs or standard operating procedures and the documentations, and then there should be setting up of the quality assurance system for the production as well as for the services QA system, and then there should be communication in the internal system as well as in the external system, and proper hygiene and sanitation should be maintained. Proper training should be given to the personals who are handling, and then periodic evaluation should be done, and if there is any update, that should also be done, and all the process should be documented, and this all comes under the role of organization. So the HACCP plan, uh, it must be updated regularly also, and CCP documentation each hour, it's not possible, so it's incorrect. So B is incorrect, all are true. So is it clear, this question, to everyone?
Yes, ma'am.
Yes. So let's move to the next question. Which of the following statements is true with respect to gluten-free products? So gluten-free pasta is traditionally made from rice flour obtained from long grain varieties as they contain high amylose. Then statement B is saying cereal germination can increase immune stimulatory gluten peptide and thereby diminish its toxic effects. And statement C is saying sourdough is basically a mixture of flour and water fermented with a starter culture of yeasts only. And option D is saying HP is in the range of 10 to 100 megapascal, can be applied in modifying the functional properties of proteins and starches in gluten-free flours. So which statement should be correct?
Option C, ma'am.
Option C.
No, ma'am, not option.
Not.
No, ma'am, not C.
Not C. Let's see in the chat box. Maybe we can find answer there. D, B, C, A. Oh, okay. A is mainly A. Yeah, A, A is there. And D also. D and A. O- Okay, let's see. Hmm.
Option D, ma'am.
Option A is correct. I'm so sorry. D is not correct. Let's see. Let's understand why. So basically, flours and starches of gluten-free cereals, they consist of rice or corn and pseudo cereals like the amaranth, buckwheat, millets or quinoa. They are traditionally gluten-free pasta. It is made from rice flour. Usually, flour is obtained from long grains and is preferred because it is having high amylose content. When rice flour is used as the only ingredient for pasta production, is required to add additives or particular processing techniques to modify it in a suitable way, uh, the properties of the starch or proteins which we are using or the macromolecules which we are using to get the final product and final structure of the product. So this is true because gluten-free pasta, it is from, like, traditionally it is made from rice flour with the long grains because they are having high amylose. Next, let's see, B, C and D is not correct. Why? Because in novel technologies for gluten-free product development, we use enzymatic approach. Like there is usage of proteolytic enzymes to detoxify gluten by cleavaging the peptide bond next to proline and glutamine residue with the action of PEP. So it is an enzyme which is prolyl endopeptidase, and it break this proline and glutamine, the bond next to it, and forms these residues. So unlike human gastrointestinal protease, PEPs, they can degrade gluten to amino acid or non-toxic peptides less than 9 amino acid residues. And in cereal germination, there is the degradation of immune stimulatory gluten peptides and thereby diminish its toxic effects. So in ferment germination, there can be the role of lactobacillus, lactic acid bacteria or lactobacillus, which are well known to process a complex protease system which can hydrolyze various proline residues of gluten. These enzymatic approaches are reported to be good step in reducing the toxic effect or the allergic effect of gluten. So, by the help of different enzymes like proteolytic enzymes and cereal germination, the gluten can be broken down and its toxic effect can be reduced. But here option B is saying that cereal germination can increase immune stimulatory gluten peptides and thereby diminish its toxic effects, which is, if we see, it degrades. So this option is not correct. Next is sourdough is a mixture of sourdough is a mixture of flour and water fermented with starter culture of lactobacillus and yeast. So in sourdough fermentation, it enhances dough property, it improves volume, texture, flavor, and nutritional values. And in this image, we can see that what is the sequence of protein degradation during fermentation of sourdough. So this is flour, and if we see, it is release of peptides. So in the flour, lactobacillus culture will be there, and there will be gradual acidification of the dough, then activation of endogenous enzymes, and then primary proteolysis of the flour, and finally the release of peptides. This lactobacillus culture, it hydrolyzes by intercellular peptidase of lactobacillus species, and further it converts into the peptides. Finally, the viscoelasticity property of the dough can be changed and there can be formation of some of the flavor compounds or flavor precursor compounds. Option C, in option C, it is saying that it is a mixture and water fermentation with a starter culture of yeast only, which is wrong because it is a starter, it is having starter culture of LAB and yeast. So this option C is also not correct. And last is the high pressure processing. It can be one of the promising technology which can be used to improve the gluten-free dough structure in HP. It is in the range of 100 to 1000 megapascal, which can be applied to modify the functional properties of proteins and starches in gluten-free flours. The high pressure, it causes starch to swell and gelatinize without the disruption of granular integrity, which is normally seen during the heating process. The effect of the extent of swelling depends upon what is the pressure applied, or the for how much time we are applying, at which temperature, what is the concentration and type of starch. It depends. So in D, it is saying it is in the range of 10 to 100 megapascal, which can be applied to modify the functional properties of starch and the protein in gluten-free flours, whereas its range is 100 to 1000 megapascal. So this option is also not correct. Is this question clear to everyone? Why A is correct and B, C and D are not correct?
Yes, ma'am.
Yes,
Yes?
ma'am.
Yes, ma'am.
Yes, ma'am.
Okay. So let's move to next question. Which of the following is a CCP in the production of fortified blended food by extrusion? So statement A is saying checking the length of the mixing cycle to obtain homogeneous mixing in the blending of maize or corn and soya in a horizontal mixture. B is maximum of 10% moisture content of extrudate. C, grinding of maize soya blend through 6 to 8 screen. And D is constantly operating fan during cooling of extrudate. So which is a CCP when we are making fortified blended food by the process of extrusion? Any guesses?
Ma'am, option B.
Option B,
Option C.
option C. Okay, let's see. B is correct, which is a CCP, because checking the length of the mixing cycle to obtain homogeneous mixing during blending of maize and soya in the horizontal mixture, which is A. Then grinding of maize soya blend through 6 to 8 screen, which is C. And then constantly operating fan during cooling of the extrudate to remove steam and condensate that can encourage microbial growth are considered, this statement D, they are GMP. These three are GMP. So these come under good manufacturing practices in the when we are producing fortified blended food by extrusion. While when we limit the moisture content extruded of the final product up to 10%, then while we are producing fortified blend, it is basically a critical control point or CCP, because if there is more than 10% of moisture content, there can be growth of microorganisms, and finally it can affect food stability or food safety. It can cause food safety hazard. So that's why, or biological hazard in the food product. So it is CCP, which is critical control point here. So option B is CCP in this process. Is this question clear to everyone?
Yes, ma'am.
Yes.
Ma'am.
So let me take today's attendance. Okay. Let's move to next question. Match the types of algal genus in column 1 with its characteristics pigment in column 2. So we have in column 1 algal genus, which is spirulina, Hemometacoccus, and then C is cholera, and D is porphyra. So in D, in option in column 2, they have these different pigments like lutein, astag, uh, astaxanthin, and then psychoerythrin and psychokine. Kinin, I'm sorry, I'm not pronouncing it correctly. So, which is matching to which algal genus is producing which pigment?
Option C.
Ma'am, option D.
C
Option
and D.
D, ma'am.
Option C and D. Is there anyone who is having another option? So D is correct, as we can see in this table also, that hemaco hematococcus, it is here. So it is producing astaxanthin as its pigment, and then cholera, it is producing different, it is an algae which is producing lutein, chlorophyll, and beta carotene, and then spirulina. Yeah, this is a spirulina. So it is producing these three compounds: beta carotene, chlorophyll, and psychocyanin. And this is porphyra, which is producing chlorophyll and psychoerythrin. So option D is correct. This is spirulina. First is hemo. This this is this, right? Yes. And then spirulina. This one. This is lutein. Last is this, right? So is this question clear to everyone of why option D is correct?
Yes, ma'am.
Yes. Let's move to next question. Blank type of agglomerates appear when droplet of high moisture collides with large amount of fine during agglomeration. So there are different kind of agglomeration. Uh, so based on how they appear, they have been given some name like onion, raspberry, compact grape, and loose grape. So we need to tell which type of agglomerate appears when droplet of high moisture collides with large amount of fines during agglomeration process. So this agglomeration process basically occurs during fluidized spray drying process. This is a hint for all of you. So let's discuss the answer. So raspberry is the answer. So the appearance of agglomerates, basically it appears in fluidized spray dryer. Is generally it is classified as these onion, raspberry, and grape. So because it is, this classification is based on like by having proper process condition, like it depends on temperature or the particle viscosity, and because of that structure of agglomerates may vary. So we can see here is an onion, it is an a raspberry, and this is compact grape, and this is loose grape. So onion, it is this form when small droplets of high moisture contact with fine particles and then spread over the surface. So small droplets of moisture, when there will be very small moisture droplets and it contact with the fine particles, which is also very small or fine particle, and because of that onion, this kind of a structure forms. And in raspberry it forms when a large droplet of high moisture it collides with large amount of fine particles, fines or very fine particles. So fines it adhere to the surface but do not penetrate the droplet, so it will be like on the surface. And then grape it formed by collision of similar quantities of droplet and fine particles. So it may form compact structure and it may form loose grape structure. So here we can see in the more clear image that in onion structure, so there is in the core there is powder particle or the fine powder particle, and there will be different these are the atomized droplets of liquid or the moisture outside it. And in raspberry, droplet will be inside and fine particles will be on the surface. And in the grape structure, it is like the in the equal proportion, the water droplets and fine powder. So it is like quite compact, and if this is loose structure. So when we go this side, so powder volume or bulk volume it will going to increase because this is the loose structure. So more pores will be there in this structure, so more bulk volume. Whereas in onion structure is having more mechanical strength or stability, while in low or loose grape structure the mechanical strength it is low. So higher moisture content droplets result in compact structure and high mechanical strength, and the optimum dissolvable power dissolvable powder are generally obtained between compact and the loose grape region, and they have better solubility in between these two structures. Is this question clear why option B is correct?
Yes, ma'am.
Yes, ma'am.
Yes? Everyone, is it clear?
Yes, ma'am.
Yes? So let's move to this question. Which among the following is the correct sequence of steps for pasta production? So mixing, hydration, extrusion, and drying. Which is the correct sequence of pasta production?
Option A, ma'am.
Option A
A, either C.
or C. Let's see everyone's opinion. B, C, D. Mainly C and D. Let's see. So C is the correct answer. Pasta is basically prepared in 4 steps, which includes hydration step for incorporation of water in the dough. First, we will add water in the dough, so hydration, and then mixing of the dough. So this is mixing. Then, because of mixing, it leads to the formation of gluten network. And finally, the shaping and cutting, which is done by extrusion process. And at the end, there is drying process. So drying of the extruded to get pasta. So option C is correct. Is this question clear to all?
Yes, ma'am.
Yes. Thank you. Let's move to, okay, let's see this and then we will move to next question. So we will have in even flow diagram, we can see that there is flour, in that there is water we are adding in the initial state, which is hydration, and second step is the mixing. So we are having all these flour and the water, and whatever is the gluten alternative we are taking, and additives, which is xanthan gum we are taking, and then we are mixing. So second step is the mixing, and then passing it through 200 mesh size. After that, extrusion process is there. So where we will going to maintain some particular RPM of the feed and the extruder, and temperature is also maintained. And then ribbon dye in this it is third process. And then finally we will dry to 12% moisture content and pack, and we can get our final extruded pasta, which we can see in here in this diagram. So final drying is up to 12% at max. We are doing. This is proper sequence of pasta production. So we have discussed many concept based questions. Let's solve one numerical problem. Uh, of This is the first numerical problem of the today's class. So please try to solve in with the paper and pen. So let's see the question. The energy required to reduce the particle from a mean diameter of 10 mm to 3 mm is 11 kilojoule per kg. Using Rittinger's law, calculate the energy requirement to reduce the same material from a diameter of 3 mm to 1 mm. So we know that in when we are making powders or we are doing crushing and the grinding operation, we have some laws associated with that. So one law out of them is Rittinger's law. So there are actually three laws. Rittinger law, which is in this question. And then second is Kick's law. And third is Bond's law. So this question is about Rittinger's law. So we have given the mean diameter is reducing, like from initial it is the particle is of 10 mm and then final diameter is 3 mm, and the energy is given that it requires this much of energy. So we need to calculate if the particle size is 3 mm, then if we want to reduce it to 1 mm, then how much energy should we apply? So please try to solve this question. Let's understand the solution part. So Rittinger law is basically, it says that energy required to reduce the size of a solid is directly proportional to the increase in surface area. So if we are applying some of the energy to reduce the particle size, so it will be directly proportional to the increase in surface area. So it depends on how much surface area is increasing. So based on the surface area, we have to apply we need that much of energy for crushing or grinding. So in the equation form, this says that E is equals to, E is the energy required, is equals to K, which is Rittinger's sorry. So E is equals to energy required equals to K, which is Rittinger's constant, into 1 upon xp. Xp is the length of the product, and which is final product, minus 1 upon xf, which is length of the feed material. So in this equation, if we put E equals to K 1 upon xp minus xf, so in first case, it is given that 11 kilojoule per kg of the energy is required. So 11 energy is equals to K, which is Rittinger's constant, into 1 upon final product length, which is 3 mm. So 1 upon 3 minus xf, which is 1 upon xf, which is feed's length. So initially we are giving 10 mm as the length or mean diameter. So 1 upon 10. So when we solve, so LHS will be 11, which is energy required, equals to K into, if we solve this, then it will be 10 minus 3 by 30. So K, which is retention constant, is equals to, this can go here. So 11 into 30 divided by 10 minus 3, which is 7. So 11 into 30 divided by 7. So when we solve this, it will be 33330 divided by 7, which is equals to 47.14. So we now know what is the Retinger constant value. Now we can calculate what is the energy required when we reduce the particle from 3 mm to 1 mm. So E is equals to, it is energy required is equals to Retinger constant, which we just now calculated, 47.14 into 1 upon XP, which is the final product dimension. So we are reducing this time particle from 3 mm to 1 mm. First we reduced 10 mm to 3 mm and then 3 mm to 1 mm. So 3 mm is the feed now and 1 mm is the final product dimension. So 1 upon 1 minus 1 upon 3, 3 is feed dimension. So 1 minus 1 upon 3, this is 1 minus 1 upon 3. So it is 2 by 3, which is this. So 2 by 3 into 47.14 will be 31.43 kilojoule per kg, which is coming in this range, 31 to 32 kilojoule per kg. So option B is correct. Is this question clear to everyone?
Yes, ma'am.
Yes.
Yes? Let's move to next question. A pulse mill grinds Bengal gram having volume, surface, mean diameter from 2 mm to 100 micrometer. So which is the ratio of retingers to Kick's constant? As I just now discussed that there are three laws related to when powder is grinding or undergoing crushing. So Rittinger's law, Kick's law and Bond's law. So here we are milling Bengal gram from volume, surface, mean diameter 2 mm to 2 mm to 100 micrometer. So we need to tell the ratio of Kick's Rittingers by Kick's. Please calculate. So 100 micro means 0.1 mm. Let's see solution part. So we just now discussed Rittinger's law. So we know Rittinger's formula also. E is equals to K. Here we are representing constant as Kr because we need two constants here. So E, which is energy required to crush or grind, is equals to Kr, which is Rittinger's constant 1 upon Xp by 1 upon Xf, which is final product dimension minus 1 upon final product dimension minus 1 upon initial product length. So E is equals to Kr 1 upon, as we discussed here, it is 100 micro, 2 mm to 100 micro we are reducing. So 100 micro can be 0.1 mm. 100 divided by 1000, right? Because to convert in mm. So it will be 0.1. So 1 upon 0.1 minus 1 upon 2. So this is because initial particle size 2 mm. So 1 upon 2, which is equals to Kr equals to, if we see this, it will be 10. So 10 minus 1 upon 2, which is 0.5, will be 10 minus 0.5 will be 9.5. So 9.5 Kr. So Kr, which is Rittinger's constant, is equals to E, and this will go here. So it will be E by 9.5. This is equation 1. Is this clear till this point? It's clear, right? Can we move to second part, which is finding Kick's constant?
Yes, ma'am.
Yes.
Yes. Yes, ma'am.
Yes, okay. Let's see what is Kick's law. So Kick's law is energy required to reduce the size of a material is proportional to the logarithmic ratio of the initial to final particle size. So it is logarithmic ratio. So E is energy required equals to K which is Kick's constant, ln which is logarithmic ratio of initial feed, which is initial product dimension, divided by final product dimension, which is xp. So this is Kick's law. So E is equals to Kick's constant, energy is equals to Kick's constant into ln and xf, which is, we are saying that our particle is going to reduce from 2 mm to 0.1 mm. As per this is given in the question itself, right? So initial, which is xf, is 2 mm. So 2 divided by xp, which is final product dimension, which is 0.1. So 2 divided by 0.1 is 20. So E energy is equals to Kick's constant into ln 20. ln 20 is 2.995 into, this is Kick's constant. So Kick's constant is equals to, this will go here. So E by 2.995. This is our equation second, right? And from equation 1, we calculated Rittinger's constant is equals to E by 9.5, right? So in equation 1, we will going to put the value of E, which is energy, from equation 2. So this is equation 2. It is energy here. E is equals to 2.995 into Kick's constant. So we are going to put value of E in equation 1, which is Rittinger's constant equation, and we can find the relation. So Kr, which is Rittinger's constant, is equals to E. E means 2.995 into Kick's constant. So 2.995 into Kick's constant divided by 9.5, which is this. So Rittinger's constant, this, and we are dividing by Kick's constant divided by Kick's constant, which is the ratio we needed to find out, right? So is equals to this 2.995 divided by 9.5, which is 0.3153 mm or 315.3 micrometer. which is nearly around option D. Is this question clear? This calculation clear to everyone?
Yes, ma'am.
Yes? Let's move to the next question then, if it's clear. So, we have discussed what is Kick's law, what is Rittinger's law. Anyone in anyone if they know about Bond's law, what is Bond's law? Please let me know. Bond's law? Anyone? Third law? Let's see what is Bond's law. So Bond's law, it states that the work or energy needed to crush a material is proportional to square root of the surface to volume ratio of the particle. So it is in this, it is under root of square root of the dimensions. So here E is the energy, input energy, M is the mass of the material, E by M is energy divided by mass. Or it can be if we have kilojoule per kg in question, then we can use that as here in this part equals to kb, which is Bond's constant, and then 1 upon under root dp, which is final product size, minus 1 upon under root df, which is feed size or input particle size. So this is Bond's law. So if the question comes, you can answer Bond's law also. Let's move to this question. Next question. Rice is milled from 7 mm to 400 microns using a 10 HP motor. Using Kick's law, calculate the HP requirement of motor to reduce the particles from 7 mm to 150 microns. Please, you solve this question because we have solved such type of questions before. This is Kick's law question. We need to uh, tell the HP requirement, power or energy requirement we need to calculate. Before this, we calculate such question. Please try to solve this question. Let's solve this question. So we know Kick's law that E is equals to Kick's constant ln particle dimension of feed divided by the dimension of the final product. So it is giving that 10 HP motor we are using, means 10 horsepower we are applying. So 10 energy E equals to 10 equals to the Bond's constant, which is Kk ln. We are reducing particle size from 7 to 7 mm to 400 microns, which is 400 divided by 1000 is equals to 0.4 mm. So initial product dimension is 7 mm. So ln 7 divided by final product is 0.4 mm. So ln 7 by 0.4. So ln 7 by 0.4 is ln 17.5 into, this is Kick's constant. ln 17.5 is 2.86 into Kick's constant. So Kick's constant, this is equals to 10, this divided by 2.86, which is 3.494 is the Kick's constant. Now we are seeing how much energy is required to decrease from 7 mm to 150 microns, which is 150 divided by 1000 is 0.15 mm. So again we will apply this Kick's law. E is equals to Kick's constant Kk, which is, we just now calculated this, 3.494 into ln xf, which is feed dimension, which is 7 mm. This, so 7 divided by final product dimension, this time it is 0.15 mm. So 7 divided by 0.15. So ln 7 divided by 0.5 is ln 46.67, then into Kick's constant, which is 3.494. ln 46.67 is 3.843 into Kick's constant, which is 3.494, which is Kick's constant. So when we multiply this, they will be, their multiplication will be 13.43 horsepower of energy we will going to require. So the 13 to 14, this is D option, 13 to 14. HP motor is adequate to grind the particle from 7 mm to 150 microns. Is this question clear to everyone?
Yes, ma'am.
Yes? Okay, let's move to next question. Which of the following is good manufacturing practice for food processing equipment? Less disassembled parts as possible in processing, flat bottom vessels, round edges of the vessel, all of these. Which is good manufacturing practice for equipments? Please, anyone, please share your opinion. It's very simple question. Which one?
A.
A. So we got C and A as our answers. C and A. Okay. Let's see. C is the answer. So in GMP equipments, we should have proper SOP should be available for every equipment. First point is this. We should have every proper maintenance of SOP for every equipment, and then more disassembly component should be there because it will be easy for cleaning. So this less disassembled is wrong. More disassembled parts should be there so that we should have more able to clean properly. Then flat bottom vessels restrict flow of liquid out after processing, and the remaining residual fluid in the vessels may harbor microbial growth. So if there is this flat bottom vessels like this or like this, it will be difficult to flow out the liquid, and because of that there may be possibility of development of microbes, and it will not be good. But round bottom vessels, it is easy for cleaning purpose than the vessels with sharp bottoms and is a GMP. Good manufacturing practices, we should have round edges of the vessel like this so that it is easy for the liquid to flow and there will be no microbial growth. This can also be possible like in this also. It is easy to this kind of bottom in this also, it is easy to flow the material. And all the equipments, they should also be covered and protected so that there will be prevention of contamination, no contamination will be there. So this is good manufacturing practice that we should have a round edges of the vessel. Is this question clear to everyone?
Ma'am.
Yes, let's move to next question then. Okay, there are some other also like in the slide in the lecture video, some other requirements also they are given uh, for GMP practices of the equipments, like there should be installation at a distance 60 centimeters should be away from wall and 30 centimeter from the floor, and they should be cleaned and disinfected after every maintenance, and the interior surfaces in the contact of the product they should be able to self-emptying, and final product handling station must be fitted with metal detectors of appropriate sensitivity. So all these are cover under GMP of the equipments. So let's move to this question, which is a solving question, numerical question. So a rice polisher was used to polish brown rice. A relationship between percentage head yield and percentage bran removed is given y and x respectively, and there is relation which is given that percent head yield is equals to 95 minus 2 into percent bran removed, where x is in the range of 5 to 20. Calculate the broken rice percent if degree of polish is 6%. So the relation is also given, so it's easy to calculate, right? Please try to calculate this question. This is very basic question, just based on some basic reasoning, because formula is also given y is equals to 95 minus 2x. Okay, let's see solution. So brown rice, when we have brown rice, we polish it and we get brown white rice, and we get extra bran separated out of that, right? This is the normal procedure when we are doing when we are polishing the brown rice to white rice. So in the question it is given that the percent head yield, which is y, it is equals to 95 minus 2 into percent bran removed. So 95 minus 2 into 6. Why we are taking 6%, 6 as here? Because percent bran removed, which is degree of polish. That when we are doing brown rice to white rice, there is 6% degree of polish. So 6% is the bran which is removed. So 6. So 95 minus 2 into 6. So 2 into 6 is 12. So 95 minus 12 is 83. So 83% head rice of white we are getting. So finally we need to calculate broken rice percent. So broken rice percent is equals to 100, which is the total, minus 83, which is white rice which we are getting as final product, minus 6, which is brown, this bran we are getting, and this is white rice long grains, and then we also get this is bran which is the waste material, which is 6%, and then we also get one fraction which is broken. So we get in total 3 things from brown rice. So the broken one will be 100 minus white rice, or the head yield we are getting minus 80. So which is 83. So 100 minus 83 minus 6, which is degree of polish, or the bran, which is percentage of the bran which is removed. So 100 minus 83 minus 6 is equals to 11. So we are getting here, which is 100 minus 89 equals to 11. So 11 is the broken rice percent. Is this question clear to all?
Yes, ma'am.
Yes?
Yes, ma'am.
Yes, okay. Let's move to next question then, which is about probiotics and prebiotics. So which among the following statement is a true? So we need to find true statement with respect to probiotics and prebiotics. So first is, probiotics are the non-digestible food compounds that help the growth of beneficial bacteria in the gut. Second statement is, oligosaccharides, resistant starch, etc. are the prebiotics. Then third statement is, probiotics are the live beneficial microorganisms added to the diet. And fourth statement is, yeast, lactic acid bacteria are prebiotics. So we need to tell which is true. Only first and second, only 3, and 2 and 3. Mostly everyone is saying D and B. Let's see solution. D and D means 2 and 3 or 1 and 2. Okay. So D is correct. 2 and 3 is true. So let's see why. Prebiotics, they are basically the non-digestible food components that help the growth of beneficial microorganisms in the gut. So we can see here in the image that prebiotics are basically the non-digestible food components which help beneficial microorganisms to grow. They can be like carbohydrates, like oligosaccharides, resistant starch, etc. And then probiotics are the one which are the live beneficial microorganisms which are added to diet like yeast, lactic acid bacteria, etc. And their benefit is that they can enhance immune function and reduce blood cholesterol level. So if we see here, option second, which is saying oligosaccharides are resistant starch are prebiotics. So it is very clear from this that oligosaccharides and resistant starch are prebiotics. So second is correct. Then 3 is probiotics are the live bacteria which are added in the diet. So probiotics is the live bacteria which are added, which is also correct. Fourth is saying lactic acid bacteria and yeast are prebiotics. But here we can see it is probiotics. They are probiotics. So it is not correct. Also, in statement 1, probiotics are saying non-digestible food components, whereas probiotics are the live organisms and prebiotics are the non-digestible food components. So this option is also, first statement is also not correct. So D is correct answer. Is it clear to everyone?
Okay, ma'am.
Yes,
Clear?
ma'am.
Yes.
Yes? Okay, let's
Yes,
move
ma'am.
to next question. Yes. Thank you. Let's move to this question. What among the following is a pseudo cereal? Wheat, amaranth, quinoa, barley. Option A is 1, 3 and 4. B is 2 and 3. C is 1 and 4. And D is 1, 2, 3.
Option B.
Option B.
Option B, ma'am.
Everyone, option B.
Yes, ma'am. Option B.
Okay, you are all correct. Option B is true. So buckwheat, amaranth, quinoa. These three are pseudo cereals and they are gluten free, whereas wheat and barley they are cereals. So B is pseudo cereal, which is amaranth and quinoa. We can see here also that these are all gluten free products. In gluten free products, we use these kind of cereal flours and starches. So here also, if we see pseudo cereal, in pseudo cereal, there are these three: buckwheat, amaranth, and quinoa. Right? And these are other like conventional sources of the gluten-free products, which includes rice flour, corn flour, potato starch, cassava starch. Non-gluten cereals includes rice, maize, sorghum, millet. And roots and tubers includes cassava, yam. And legumes may include soya, chickpea, beans, lentil peas. And others can be flaxseed, chia seed, chestnut, unripened banana. They all can be used as in the gluten-free products, and they are And the wheat and barley, they are cereals mainly, which are here in this option. And quinoa and amaranth, they are pseudo cereals. So let's move to this question, which is last question for today. So let's discuss this. Match the food powder type in column A, which is like when we are grinding food powder. So it can be grinded in various dimensions. So it can be considered as dust, powder, flour, or granules. So based on the particle size, we categorize these different food powder types. So what is dust? So let's see
Option
if
C.
you, okay, option C. Let's include everyone. So
Option
I
C.
C, C or D.
Yes, ma'am.
Option A.
Okay, A, C, D, but maximum are on C, right? Let's see. Option C is correct. So there are different type food powders with which are having different terminologies based on their particle size. So powders are the one which are in the dimension of 50 to 200 micrometers, which are the finest among all. And they are like, they can be like in the milk, coffee, colorants, and their characteristic is like they are more cohesive and higher in bulk density. Then we have granules of the little coarser size, 200 to 4000 micrometer, and examples include instant milk, instant coffee, and semolina. They are free flowing and having low bulk density. Then is the flour, which is having 100 to 5000 micrometer. They can be of like cereal flours, mainly nut flours, soya flours. So they have irregular particle shape, large size, and low fluidity. Final is the dust, 5 to 100 micrometer, which is like any powder. So they have the finer than the desired size, normally fly during processing and handling. So if we see here, dust is the smallest one, which is 5 to 100 micro, and then powder, it is 50 to 200 micro. Then flour is this one, 100 to 5000, and granules can be lie in the range of 200 to 4000 micrometer. So, is this question clear to everyone?
Yes,
Is
ma'am.
there any doubt in today's session? Today's any question?
No, ma'am.
No? Okay. So today we discussed all these topics, like we discussed about functional foods and nutraceuticals, about their health benefits, and we discussed about prebiotics, probiotics, when also we discussed about algae based health foods and the like bioactive compounds or the pigments which algae produce. And then we discussed about gluten free breads and the pasta, and we discussed about gluten intolerance or the celiac disease. Then we discussed about some of the processing conditions of gluten-free bread and pasta. And then we discussed about powders and premixes, their basic introduction, which is like, which is dust, which is powder, which is granules, and like which is coarser particles. So we discussed about all these, and then we discussed also about the agglomerates, mainly which is included in the drying process, and then we discussed about their formulation also, and we discussed about GHP, GMP in the food uh, industry, and we discussed about HACCP, TQM also, and then CCP also we discussed about in today's lecture. So today is the last final session for this course and in this semester. Uh, so thank you everyone for joining, and special thanks to the one who has joined from week 1 to week 12 all the classes regularly. And I hope you please try to submit your assignment 12 on time, and a very good luck for your examinations. Uh, that's it from my side. Thank you so much. Thank you.
Thank you, ma'am.
Thank you, everyone.
Thank you, ma'am.
Thank you, ma'am.
Thank you. Thank you, everyone. I hope I was able to deliver uh, the classes. Like I tried my best. Maybe there can be some improvements, but yeah, I tried to deliver, and I hope you like my lectures.
Yes, ma'am.
Yes, ma'am.
Okay, thank you. So thank you so much for and ma'am, exam similar questions. Yes, in exams you're going to get similar questions as your assignments. So in assignments you solve many questions. So yeah, so those type of questions will be appearing for your examination. So please practice well and write your examination in a good, how much you can do in like the best of yours, and do not panic. I hope you all will write, like my good wishes are there for all of you to write good examination. Okay, so if you don't have any questions, we will leave today's class. Okay, you don't have any question. You can leave, which is not like who is not having any question. If you have any question, please be here. You can ask your doubts, or otherwise you can leave. No questions. Okay, guys. Good luck. How many numericals?
Yes, ma'am.
Okay, how many numericals? It will be the same proportional how you are solving your assignments. So it's not like that numericals won't be there. You have to do both numericals and the concepts both. And it will be mostly like how you are doing your assignment the same way. Okay,
Okay, ma'am.
what ma'am, what is lethal rate summation? Sum of lethal rate? What you want to ask? I'm not getting.
बोलो ना की बोल रहे हो
Alok, can you write your question in detail? What you want to know? Ma'am, week 2 assignment question no. 9. Is it MCQ type or paper pen type? But you need to solve, right? Week 2 assignment question 9. Week 2. Lethal rate summation means if there are many microorganisms, everyone's lethal rate, like at all the points, like if there is any container and there are, so we are taking, we want, like we are taking from different points, what is the lethal rate, and uh, like on an average we are taking, and we are taking that point as our, so that is lethal rate summation, because we are not taking at, we cannot take from just one point in the whole batch when we are going for a pilot scale. So that time we use lethal rate summation. And there can be just not one pathogen, right? There can be other pathogens as well. So we have to take the lethal rate of those as well. Just like by determining one pathogen, we cannot say that its lethal rate is the final one. So is it okay? I'm stopping the recording for today, and if you have any doubt, you please be here, otherwise you can leave the meeting.